Dear students, today we are going to see the topic biological oxidation under Unit 1. Oxidation is a reaction with direct addition of oxygen or removal of hydrogen or loss of electrons. The biological oxidation and reductions are restricted to three simple classes. One is loss of one or more electrons, loss of one or more hydrogen atoms, and addition of one or more oxygen atoms. In biological oxidations, the term exothermic and endothermic are replaced by exergonic and endergonic. For example, a substance A is oxidized to B with the release of energy, and the oxidation is coupled to another reaction in which C is being converted to D. Now, some of the energy liberated in oxidative step A to B is transferred to the synthetic step C to D in the form other than heat. This is called free energy. Thus, in exergonic and endergonic reactions, free energy is released or absorbed respectively. The principles of biological oxidations of carbohydrates, proteins and fats are: first of all, complex organic molecules are degraded into 2-carbon compound. The 2-carbon compounds are then broken down by a series of steps. In each step, 1 carbon dioxide and 2 hydrogen atoms are removed. Decarboxylation of organic acids removes carbon dioxide without any considerable change in energy. The second end product, water, arises from reduced coenzymes of respiratory chain and molecular oxygen of atmosphere with production of some energy. The first process of oxidation is by direct action of oxygen. Number of enzymes catalyze direct interaction of substrates with molecular oxygen. Depending upon the fashion in which molecular oxygen is used, these enzymes can be classified as oxidases, oxygenases, hydroxylases and hydroperoxidases. The first one is oxidases. These are electron transferring oxidases and catalyze removal of hydrogen from a substrate by directly using oxygen as hydrogen acceptor with water as a product or hydrogen peroxide as a product. If water is formed as a product, we can say cytochrome oxidase, ascorbate oxidase as examples. If hydrogen peroxide is formed as an end product, we can say examples: amino acid oxidase, xanthine oxidase, aldehyde oxidase, glucose oxidase, etc. Some of them are copper containing enzymes and oxidize a substrate by transferring reducing equivalents from it to molecular oxygen. The next class of enzymes are oxygenases. These enzymes incorporate oxygen into their substrates but are not concerned with energy production. They have two subclasses. First one is dioxygenases. These catalyze the incorporation of both the atoms of oxygen into the substrate. Example: tryptophan 2,3-dioxygenases. If it is monooxygenase, these incorporate one oxygen atom into the substrate to form hydroxyl group on it. Example: mitochondrial cytochrome P450 monooxygenase. These enzymes transfer reducing equivalents from NADPH or NADH. There is another group of enzymes called hydroxylase which also fall under this category. They are sometimes called as mixed function oxidases. Example: tyrosinase, phenylalanine hydroxylase, etc. The third classification of enzyme is hydroperoxidases. They catalyze oxidation in which hydrogen peroxide acts as hydrogen acceptor and is reduced to water as AH2 plus H2O2 gives A plus 2 molecules of water, 2H2O. A plus H2O2 gives AO plus H2O. All peroxidases found in plants and milk and catalases found in animals and plants are examples. The second type of oxidation results from loss of hydrogen. Enzymes that remove hydrogen from the substrate fall under this class and are called as dehydrogenases. They are of two types, aerobic dehydrogenases and anaerobic dehydrogenases. Aerobic dehydrogenases are flavoproteins bearing FMN or FAD as the prosthetic group. They accept two hydrogens from it on the FMN or FAD, which is thereby reduced to FMNH2 or FADH2. These can be further reoxidized by donating the hydrogen to molecular oxygen forming hydrogen peroxide. Example, we can say L-amino acid oxidase, xanthine oxidase for aerobic dehydrogenases. The second classification of dehydrogenases is anaerobic dehydrogenases. In this group of enzymes, there is direct transfer of electrons to molecular oxygen. Pyridine-linked dehydrogenases, flavin-linked dehydrogenases are coming under this category. Pyridine-linked dehydrogenases oxidize the substrate by transferring a hydride ion from the substrate to NAD plus or NADP. The second hydrogen removed from the substrate is released as free H+. In the process, the NAD and NADP get reduced to NADH and NADPH. The flavin-linked dehydrogenases are usually FMN and FAD. FMN and FAD are the two flavin-containing coenzymes that remain linked to specific dehydrogenase enzymes. In the process, the flavin coenzymes get reduced to either FMNH2 and FADH2 depending upon the enzyme. Usually FAD-linked enzymes are D-amino acid oxidase, glycine oxidase, succinic dehydrogenase, and FMN-linked enzymes are NADP cytochrome C reductase. Next, electron carrier which participate in electron transport chain apart from NAD, FAD are iron sulfur proteins. A group of quinones has been found to be present in the mitochondria. They are FES, Fe2S2, Fe4S4, Fe3S4, etc. FES, it has single Fe, that is ion, coordinated to the side chain SH groups of 4 cysteine residues. Fe2S2, it contains 2 iron atoms, 2 inorganic sulfides and 4 SH groups. Each ion is linked to 2 SH and 2 sulfur groups. Fe4S4, it consists of 4 iron atoms and 4 cysteine SH groups and 4 inorganic sulfides. Each ion remains linked to 1 SH, 3 inorganic sulfides, while each sulfide is coordinated to 3 iron atoms. The enzymes may have one or more of the combinations of the clusters. Fe2+ of a reduced iron sulfur protein gets subsequently reoxidized by donating its electrons to an electron acceptor such as CoQ or cytochrome C1. Each iron sulfur protein transfers only one electron at a time. The next major electron carrier in electron transport chain is cytochromes. These are very important enzymes which contain heme and are involved in cellular oxidation. The oxidized form of cytochrome possesses a single Fe3 plus ion and is called ferricytochrome. It is reduced to ferrocytochrome having a Fe2 plus ion on accepting an electron. Cytochromes are identified by their characteristic absorption spectra. Ferricytochromes show diffuse and non-characteristic absorption band in visible spectrum. Ferrocytochromes exhibit characteristic absorption bands called alpha, beta and gamma Soret bands. There are different types of cytochromes. We are going to discuss about those types in detail. The first type of cytochrome is cytochrome C. Since it is available in large quantities, it is best studied of the cytochromes. It is water soluble and easily extractable. It shows characteristic absorption spectra in the reduced form at 550, 521 and 460 millimicrons. Oxidized form gives two diffuse bands at 530 nanometer and 400 nanometer. The iron content of cytochrome C is 0.38%. Heme is attached with protein by means of two thioester linkages involving sulfur of 2-cysteine and apoprotein. Cytochrome C is incapable of combining with oxygen or carbon monoxide. The next type of cytochrome is cytochrome C1. Like cytochrome C, cytochrome C1 also possesses an iron protoporphyrin 9 complex heme C. It has absorption maxima at 554, 524 and 418 nanometer. This is also incapable of combining with oxygen, carbon monoxide and cyanide. The next type of cytochrome is cytochrome B. These also contain the same protoporphyrin 9 complex called heme B. It is found to be tightly bound to flavoproteins and ubiquinones in mitochondria. The ferrocytochrome B has an absorption maxima at 563, 530 and 430 nanometer. It is thermostable and not easily extractable. It also does not react with oxygen, carbon monoxide or cyanide. The last group of cytochromes are cytochromes A and A3. They are collectively called as cytochrome oxidases. This constitutes the complex 4 of the mitochondrial electron transport chain. Both possess an identical type of iron porphyrin complex called heme A. One heme group is located along with one copper ion. This heme is called as heme A. This cytochrome A functions as the anaerobic oxidizing unit. The other heme A called heme A3 is located along with second copper ion at the binding site for molecular oxygen on subunit 1 and functions as aerobic reducing unit of enzyme complex. Cytochrome A absorbs at 605, 517 and 414 nanometer, whereas cytochrome A3 absorbs at 600 and 445 nanometer. Cytochrome A does not react with oxygen, carbon monoxide or cyanide. whereas cytochrome A3 is auto-oxidizable and forms compounds with carbon monoxide and cyanide. Next topic we are going to discuss is redox potential and about free energy. I have made it clear that an oxidizing or reducing agent may exist in two forms. The oxidized form or oxidant which can accept electrons and the reductant which can donate its electrons to a substrate. Each oxidizing or reducing agent exists as a conjugate pair of electron acceptor oxidant and electron donor reductant forms. The pair consisting of the oxidant and reductant forms of an oxidizing or reducing agent is known as a redox couple or conjugate redox pair. Example: NAD plus and NADH, FMN and FMNH2, cytochrome C Fe2 plus and cytochrome C Fe3 plus. If the sign of free energy thus calculated is negative, it indicates free energy release and delta G is negative. And if delta G is positive, it indicates free energy consumed. A system having relatively more positive redox potential can oxidize another system having low redox potential. In the mitochondria, the hydrogen or electrons pass through different carriers in the sequence of increasing order of their positive potential.